Hey guys, welcome back to another video. So today, no, I'm just kidding. Um, anyway, today I'm going to go over IB Biology 1, Saw Practice number 4. Now, this is the thick boy packet that he gave us, the double-sided 2-page, which goes over the entire, basically, almost everything that you need to know for the final, well, not the final exam, but for the test that he's about to give us, which kind of feels like a final exam because he's been alluding to this test ever since day 1. But, no need to worry. I'm going to go over it with you, and I'm going to go over it in a lot of detail, so this video's probably going to get pretty long. Again, like in the last video, at any time, if you want to skip, there will be the times in the description below. Let's say if you want to go straight to problem number 2 or problem number 3, I'll have those times in the description. Also, I didn't do this for any other video. I mean, for cell practice number 3 I did this, but starting from now on I'm going to take the entire paper, I'm going to scan it on my computer from my printer, and I'm going to upload it onto YouTube, so the entire PDF is there for you, completed, to look at. Now, I know some of you might just copy it, but at least you'll gain some sort of knowledge from it. You won't gain everything, you won't be able to understand it, but at least you'll be able to know some stuff, which I'm fine with. Alright, so let's look at question number 1. Outline the process of endocytosis. Now, endocytosis is a thing that many of you may not have heard of. Mr. Varsaler, he didn't really teach it to us. Now, that's why I'm going to really focus heavily on endocytosis and something similar to it called exocytosis. I went over exocytosis in cell practice 3, but you don't have to go back to look for it. I'm going to explain it all here now. Now, he said that he's going to go over it in class, but I'm going to go over it right now. If you want, you can skip past the time, again in the description, but I'm going to explain endocytosis and exocytosis. So just in this problem, it's talking about endocytosis. And then related to that is exocytosis. Now, already from the name, you can probably guess what each of them is. Endo, kind of in, and then exo, kind of exit out. So write that, exit. Now, these two have to deal with stuff coming inside of the cell and stuff going outside of the cell. So endocytosis is the process of stuff going into the cell. And you know, instead of protein channels, what they do, you know those protein channels, the integral proteins that they usually use for stuff to go through? Let's say this is the cell membrane, and then stuff will go through there. This is a different way. Sometimes you don't want to use proteins. Sometimes it's just, you just want to conform or something, and that's what you use endocytosis and exocytosis for. So the process of moving material into the cell. I'll just say substances inside the cell. Now, exocytosis is the exact opposite. It's the process of moving out of the cell. Now, the way that this happens, that's why it's very special, is by way of vesicles. Vesicles. Now, vesicles are made out of the same stuff that our cell walls are made out of, or cell membranes. Sorry. So, if you can imagine, let's take this cell right here. Just a normal cell, nucleus right there, and part of the cell wall, if we zoom in here, part of the cell wall, let's say there's some insulin right here, right? And this insulin wants to get into the cell. Now, one way that this could happen is by endocytosis, so inside. And what it does, if we zoom into this part right here, let's say this is a cell wall zoomed in, and insulin is right here. Step 1 would be that this insulin kind of touch the cell, just touch steps just a little bit, and going from there, it would start to make like this little bubble in this actual cell membrane. So then the insulin would go in here as well, and then the bubble will start to close around the insulin, kind of entrapping it. And then eventually, the bubble will be free inside of the cell, just like that. And then it'll get released, cell membrane, and this will pop, and it'll go out, just like that. So it's going from outside the cell kind of engulfs it some more, all the way, and then it gets dispersed inside the cell. Now basically, for exocytosis, it's the exact opposite. So instead of starting like this, we start off like this. So we have some substance that we want to get rid of. It climbs into one of these vesicles like this. It joins the cell membrane, and then it opens up like this. So if you want to write that, endocytosis over here, let's write that in blue. Exocytosis over here in red. Let's do it out so we're not moving so much. There we go. Endocytosis would be going from like this. Exocytosis would be going just like that. Here to here to here to here to here. Endocytosis from here to here to here to here. And that's basically the process of endocytosis and exocytosis. Now, there are also different variations of these two processes. So, these two are basically for any large substances. So anything that's particularly big, or a large amount, so like a large amount of smaller substances. Like a lot of ions, a lot of particles, just a lot of something, or just something really big. That's what these would be called. Now, let's say you're trying to move a solid, like a big lump, like something, a solid. That, like a bacteria or something, that would be called phagocytosis. And it's basically the same, cytosis. Now, it's basically the same process as this. It's just called something differently if it's a solid. Now, let's say if you're moving a liquid or like something dissolved, it would be called pinocytosis. I don't know if I'm cytosis. Pinocytosis. That's if you're moving a liquid or something dissolved inside of the liquid. That's when pinocytosis would come in. So, endocytosis, exocytosis, entering, leaving, large or a large amount of something smaller, and then phagocytosis, something solid, pinocytosis, anything liquid or dissolved, and they basically follow these same steps. I will leave this on the screen for a second. Some people want to take pictures, get it down, have it memorized in your head. Alright, that's good. So, back to the question that we are trying to answer. Originally, we have this, right? So this was our original question. Step number 1, outline the process of endocytosis. We already went over this. Again, some people might be skipping to this point, but a brief overview is, let's zoom in so you can read. Endocytosis is the process of moving large or a large amount of material into the cell. By way of vesicles. Capital. Alright, that's endocytosis. So, let's see here. Question number 2. Explain the structure and the properties of phospholipids and how they help to maintain the structure of a cell membrane. This is going way back to probably one of the first couple of days of class. Now, if you remember what a phospholipid is, a phospholipid is one of these things. Remember? Yeah, let's start over here. So we have a say those steps. One of these things, remember? And there are a lot of these composed inside of the cell. So it's asking how the structure and the properties of it help maintain the structure of the cell. Now, if you remember, the head is hydrophilic. Sorry. Hydrophilic. Now, this means that it's water-loving, or a lot of people use the term water-loving, but I mean, you can love someone but not want to be like really close to them. I just like to use the term, it just likes to go to water. It just wants to touch water. It just wants to. Now, whenever it can, this will just want to contact water. That's why I have it in blue. Now, the opposite is true for the red part, the tail. That's hydrophobic. Now, you've heard of something like arachnophobia, the fear of. Same thing here, being scared of water. It just, again, it does not want to touch water. It's just like, nah. So, not touch. That's basically all you have to remember. Hydrophilic wants to touch water, and hydrophobic does not. Now, using these different functions, because these are the functions of a hydrophilic tail. Now, using these functions, how does that help the structure? So, if you imagine a cell membrane, it's like dot, 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 this entire thing. All of the heads will be on the outside, and then there's going to be an inside layer of heads, and then all of the tails will be on the inside. Isn't that peculiar? So these tails would want to touch each other because it's a bilayer, remember? And things that are like each other want to go towards each other. So there'll be no water in here, but there will be water on the outside because they want to touch water. So that's how it be. Outside of the cell there's water. Inside of the cell there's water. There's water everywhere. The only place where there's not water is in between here. That's why it's like that. It's because there's water everywhere. So the parts that want to touch water, the heads, are touching water, and the parts that don't, the tails, are together, so they're not touching the water. So question 2 asks, explain how the structure and properties of phospholipids help. Due to the fact of the hydrophobic and the hydrophilic, all the same properties they arrange in a circle in water. I don't know why he gave this much space. You can go into more detail, but that's pretty much all you need. Alright. In the diagram below, macromolecules are being transported to the exterior of the cell. Oh look, being transported to the exterior, exiting. Remember this whole spiel right here? Exocytosis, out of the cell. It's like this, then it's traveling out. This is exocytosis. Oh, you didn't know that. What's the name of this process? We don't have to look at any of the other things. We know it's exocytosis. Hey. Alright. Next, annotate a label diagram of the fluid mosaic. Now this, my friends, is one hell of a problem. So what we are going to do, it's going to be super rough. You can have it nicer than I'm going to do, but this is quite rough. Basically, you need maybe 1, 2, 3, 4, maybe 5, 6-ish things that you need. So let's just go through them. So 1, you need to have some kind of integral protein. Now this isn't part of the annotating. I'm just writing this out. So this is what you need to have inside of the sorry, it's not to focus. This is what you need to have inside of the diagram. So you need to have an integral protein. You also need to have a peripheral protein. You need to have a carbohydrate. Oh, I spelled that wrong. Carbohydrate. You need to have a glycoprotein attached to that carbohydrate. Glycoprotein. You need to have some cholesterol. Just carbohydrate, there's no H. You need to have cholesterol. And of course you have to have your phospholipids. So these are the 6 things that you need to have. Now this is really important that you draw correctly. So these 2, the rest are pretty much at your own disclosure. So alright, let's draw. So when you draw this, I would just draw some circles for the phospholipids. Just get those out of the way. And then I would probably draw an integral protein like this. Not to be too fancy. That's a protein. And remember, here, annotate a label diagram. So annotate meaning you have to give the function of or some sort of description, and it's labeled, so it has to have some sort of meaning. So along you go. So what is this? This is a phospholipid. Phospholipid. We've seen this many times. And what is this? This makes up the cell wall. Next is an integral protein, so let's write that. Integral protein. Now what does this do? This goes through the cell wall and goes through the cell wall. Good. Next would be a glycoprotein, so just like add some more of these. Actually, you know what would be better? This is kind of, let's look over here. I made a nicer one before. My actual one. So let's just annotate this. Let's take a second, look at this. It's in pencil, so it's kind of hard to see, but you can kind of get the basic understanding of this. So phospholipids makes up the cell, so just write like makes up. Channel proteins, here, integral proteins, they go through the cell, so through the cell membrane. peripheral proteins, they do not go through the cell membrane. Do not go through. Okay, glycoprotein is on the surface. Carbohydrates, these are mainly for recognition. Next we have cholesterol. This is for fluidity. And then we have a phospholipid bilayer. A phospholipid. You already know these. This is hydrophobic, and this is philic. Channel protein, you don't really need to annotate. Carrier protein, you don't need to annotate. Mainly, the main ones, here I'll highlight them. Phospholipids, peripheral protein, integral protein, carbohydrate, glycoprotein, cholesterol, and then this. Basically, you only need to know for the fluid mosaic. Also, understanding, it's called the fluid mosaic because these are all shifting, these are all moving, nothing here is stagnant. They're all shifting throughout the cell membrane. Alright, here. Kind of already saw it, but the next answer is A. And the reason is A, because it's really interesting. I explained this in my other video, but I'll explain it here quickly. Okay, so water is polar. You just got to get that through your head. Water will always be polar. And this is true throughout chemistry, throughout biology, throughout everything. So H2O equals polar. Just remember that. I don't know, polar bears, ice caps, water. Just any way you need, or just remember it. Water is polar. So polar substances want to attract each other. So like I said before, there's water here, there's water here, there's water everywhere inside of the cell. The only place that water is not is here. There is no water inside of these two tails, because the heads like water, the tails despise water. And if we look at it, it's asking which of the parts of the surface protein will be composed of polar amino acids. So which parts of the surface will be composed of polar stuff? So like I said before, polar stuff wants to go to polar stuff. So anything that's touching water will have to be polar. So this is touching water, this is touching water, this is touching water. That's it. It's all touching water. Same thing for the other sides. This is not touching water over here. Not touching water. Not touching water. Because these touching water, these don't have to be polar. The ones in blue do though. So the ones in blue are 1 and 2. The ones 3 and 4 are in the inside, which are not touching water. So they do not have to be polar. If 1 and 2 were polar, then water wouldn't even want to get through here because it's going to be opposed to water, just going to go away. And there's no point of having a channel if nothing can go through. Right? So the answer has to be 1 and 2. You already saw A. Question number 5 is A. Okay. Number 6. Explain the various methods. Explain the various methods cells use to transport minerals across membranes. Okay. So this is just a question on transport modes. I did a lot of notes the day that we were supposed to do transport. And remember how he gave us a packet? Well, I didn't really like that packet, so I made my own notes. Here are my transport mode notes. Now, we're dealing with the transport of minerals, so not diffusion, osmosis doesn't count because it's the movement of water. So we can just throw that out. Well, not really, but for the sake of this right now, let's throw that out. So let's just look at this super quickly. Diffusion is passive dispersion. So over here, there's a high concentration, there's nothing here. Now they're both even. That's diffusion. Any particle, well, not any particle, but uncharged particles. Osmosis is with water, so let's say less water here. The water is pretty much the same amount, but there's a higher concentration over here, less over here. It wants to go towards the higher to make it basically equal. Facilitated diffusion uses channels. So like channels go through, and it's still going from high to low. So now it's even, it's going through here. Active transport is where it kind of gets super tricky. So it uses energy, and it goes against the high to low thing. It goes from low to high. So let's see, this kind of has a higher concentration at the beginning, this kind of has a lower concentration, and it's using energy, ATP, as you can see here, and it's going to an even higher concentration and an even lower concentration. So ATP, it needs a concentration gradient, it needs membrane proteins, it needs ATP. Facilitated diffusion, it needs a concentration gradient, it needs membrane proteins, but it does not need energy. Osmosis needs concentration, generally doesn't need membrane proteins, but uses in some situations, and it doesn't use energy. Diffusion just needs a concentration gradient, doesn't need membrane proteins or ATP. So if we summarize all of that into this, so we have diffusion, this just needs, so explain. So it's just from high to low. High to low, and it does not need energy. So no ATP. Facilitated diffusion. Facilitated. It also goes from high to low, but it uses proteins to accomplish that. So it needs proteins, but again, doesn't need energy, so ATP, no. And then for active transport, that's when you need the energy and you need the protein. So active transport. And it also goes from low to high, so it's opposite. It needs proteins and it needs ATP. There you go. So also here, let's write no, so protein. Good. Oh, sorry, couldn't see that. Diffusion goes from high to low, no ATP, no proteins. Facilitated diffusion goes from high to low, needs proteins, not energy. Active transport goes from low to high, proteins, ATP. Alright, oh, now going on to the next paper. Question number 7. Which of the following is required for osmosis to occur? Oh, here, let's bring this back. So let's look over osmosis for a second. Movement of water across a semi-permeable membrane from high to low. It needs a concentration gradient. It needs membrane proteins, but it does not need energy. Alright, let's go through this. So, osmosis to occur, does it need an enzyme? No. Oh, one of these questions in the IB exam, I know this from a lot of seniors, you're going to need to use the tool of eliminating the answers that aren't correct. A lot of times you won't know what the correct answer is, but a lot of times you will know what the correct answers are not. So right now we know that it's not going to be an enzyme. So we got that. A fully permeable membrane, that's on the roof here. We don't know if that is, so let's leave that alone for now. ATP, we know it doesn't need energy because it's osmosis, right? So it's not going to be energy. Zoom out so it stops fizzing out. And then a solute concentration gradient. Now that's interesting, right? That kind of makes sense. That also kind of makes sense. But if we think about it, a concentration gradient, we need that. We need a concentration gradient for osmosis. And a fully permeable membrane, that, if we think about it a little more, doesn't osmosis need a semi-permeable membrane? Okay, so it can't be B, but we know that it needs a concentration gradient for sure, so it's D. Alright. Explain four functions of membrane proteins. Alright, so getting into this for all the people, because I know a lot of people are going to get to this point. This Aristotle, he told us to use jet rat. This thing. Now, I really don't like this. I mean, you can use it if you want to. I'm not going to. This is confusing. Like, J stands for connecting to, like, that doesn't make sense. And A stands for, like, anchorage. Like, that just doesn't make sense. So I'm not going to use jet-rat. I'm just going to cross that out right away. What I use is something called crater. Now, I'm not going to remember it as crater because that's kind of lame, I think, personally. So you just got to remember it as cool raptors attack. 2 C's. Thick. Now you got to use 2 C's. That's the important part. Elephants. Regularly. Cool raptors attack thick elephants regularly. Now, kind of want to know what these stand for as well. Now, these are super easy because these words are associated. So, like A, pretty easy to remember. So, C is to connect. R is for receptors. A is for attachment or to attach. T is for transport. E is for enzymes. And then R is not going to be receptors. It's going to be recognition. Rewrite that. Recognition. Now, connection, receptors, attachment, transport, enzymes, and recognition. Now, I'll go through each of these. So, for the functions of a membrane protein, one of the functions is to connect. So, it's to connect one cell to another. It's not to connect like a cell to, let's say, the surface of something. It's connect one cell to another. Just join. Now, that's different from attach, because attach is from taking the cell and attaching it to a surface. That would be attachment. Connection is 2 cells together. Alright, so next is receptors. So on the edge of the cell, the membrane protein, you'll have receptors, or like remember those carbohydrates that are attaching from those glycoproteins? That would be the receptors, which signal other, like, hey, look, this is my cell, this is me. That would be a receptor. Attach, I already went over, when a cell links to the surface. T, transport, is, we did a whole unit of that. It's osmosis, facilitate transport, diffusion, active transport, all of that. Enzymes is for the metabolism of the cell, or in the outside surround, not inside of the cell, but the outside of the cell. So like stuff going on happening, because our bodies isn't just our cells, it's the stuff in between our cells. That's what the enzymes are for. And then recognition. Have you ever heard of blood types like A, B, O? That has to do with the actual membrane proteins on the surface of our cells, of our red blood cells. They help with recognition. And that's why you have different red blood types. So, that would be cool raptors attack thick elephants regularly. So, explain 4. I'm just going to pick the easiest ones, I think, to explain. So, that would be to connect 2 or more cells together. And then attachment, attach cells to a surface. So that's 1, 2, 3, I would say transport, to transport materials. And then finally 4, let's say recognition, for cells to be recognized. Now, for question 8b, distinguish between active transport, passive transport, diffusion, and facilitated diffusion. So here, so diffusion, again, passive dispersion of molecules. So going from a high concentration, nowhere, to an equal distribution. Osmosis, movement of water. Facilitated diffusion, using the use of protein channels across a high to low gradient. So high concentration over here, low here, eventually equalize, and then active transport from the area of low to high, again making the difference greater. If you want to summarize that, active transport uses energy, so let's say ATP. Passive transport does not use ATP, no energy. Diffusion, also no ATP. Facilitate diffusion, no ATP. If you want to distinguish these, this uses proteins. This also uses proteins. This does not use protein. Oh, this does not use proteins, but this also uses proteins. Distinguish these. So passive transport and then facilitated diffusion. Facilitated diffusion uses proteins, but the proteins are open. They can't close, they're always open. For passive transport, the proteins actually open and close to let different ions through. It's still going from high to low, but it's doing it in, let's say if it wants to stop it at a point like at a certain amount, it can stop it. So it's still going from high to low, just like all of these except for active transport. But the proteins can stop. Good. Alright, number 9. Which ratio limits the size of cells? We did a lot of this. So again, it's the volume to surface area ratio. Keep that in mind. The rate of metabolism to mass. We never really talked about mass and metabolism, so it's not going to be that. Surface area to volume, that's where we want. It's not going to be mass, it's not going to be surface area to mass, it's going to be B. 9 equals B. So the diagram shows a model of a biological membrane. Here is a biological membrane. What do labels I through V illustrate? And give a function of each. So we're looking through I through V. I, 1, 2, 3. He didn't really give a 4 or a 5, but we can go back to my old thing here, and you can just look at this. So phospholipids. So this would be a carbohydrate. And these have for recognition and a multitude of other things. This would be an integral protein because it goes throughout the entire cell. Integral. Just spell it out. Simple. Integral protein. 3 is just a phospholipid. Now he's going to use more than these 3. I think he forgot to label the others, so just look at this again. Leave that up there for a second. Okay. 11. The diagram below shows the structure of a cell. So state the names of 1 through 3. 1. Zoom in. Oh, it's already zoomed in. So 1 is pretty clearly the hair. If you zoom in, let's see if it zooms. There we go. It's the cell membrane. As you can see, it's pointing to the thinner portion there. So it's going to be the cell membrane. Number 2, that's where it gets tricky. He hasn't really taught this as well, but this thing, it's called a mesosome. Now, This, here, let me spell that for you. Mesosome. Now this thing is to help increase the surface area. This helps increase the surface area of the cell membrane so that it helps aid in the distribution or the diffusion of gases and waste throughout the cell. Basically increases the surface area to volume ratio that we were talking about. It makes it have a bigger size, but still relatively efficient, which is kind of weird because bacteria are kind of small, basically smaller than normal human cells, but they do it. Whoa, guys, that was crazy. Okay, so quick side note, my 512 gigabyte memory card just got full, so I had to go to my computer and delete some of my old videos to just make this for you guys. But anyway, where was I? Here. Calculate the actual. Oh no, I wasn't finished with this. Number 3. Number 3. This. Oh, this is just the cell wall. You can tell because it's like pretty thick. Alright, and now B. Calculate the actual length of the cell showing your work. Alright, so when I do a problem like this, what I normally do, and I think everyone should do, if they don't give you the length, you should get the length. You have to trust me, but you have to trust me on this, but this portion is 9 centimeters. 9 centimeters. So let's put that into here. And they also give us the magnification time, so we should keep that into consideration. 9 centimeters. Now this is 9,000 times magnified. Now let's move this into micrometers. So remember that he gave us a sheet that goes from, here it is, the metric staircase. So we're going from centimeters to micrometers, so that'll be a 1, 2, 3, 4. So we're moving it 4 to the right. So we just take 9, and then we move the decimal places 4 to the right. 1, 2, 3, 4. 9,000. So 9 centimeters equals 90,000 micrometers. Now this is magnified 9,000 times. Oh, that's cool. They're both the same. Now, if you know a little trick like I know, then you know that the answer is 1. But let's actually figure it out like let's say we didn't know what it was, because 9,000 divided by 9,000. So remember the formula that he gave us? Let me zoom in so you can focus on the work. He gave us the formula. The magnification equals the image over the actual. Now, we're trying to find what the actual is, right? So, let's try to get this by itself. So, let's multiply it on both sides. This cancel out. And then we're left on this side, we're left with A times M equals I. We're not looking for I, we're looking for A. So, let's divide this M. Let's divide this M on both sides. This cancel out, and then we're left with A. So the actual is left the image over the magnification. This is the formula that we're going to want to use. Right there. So the actual, the image. What is the image? What is this? This is 9 centimeters, which we converted to 9,000, 90,000. 90,000. And then the magnification, what's the magnification? 9,000 times. So we can already see all these cancel out, and then A just equals 1. There you go. Now, if you think about it, if something is 9,000 and is being multiplied 9,000 times, that would equal 1. So let's say this sheet of paper that I have here. Let's say this sheet of paper. This sheet of paper is 2 meters. It's obviously not. Let's say it's 2 meters. Now this would be a meter, and then this side would be a meter if we cut it in half. Now, this is 2 meters, right? But let's say this signifies 1 meter. So this is 1 meter. what would 2 meters be then? It would be half of it, right? So 2 divided by 2 would be 1. Basically the same principle here. When you have basically the same magnification and the image, they just divide each other and they equal whatever it is. So I would go about this problem starting from m equals I over a. Try to figure out what you're getting to, let's say I or an a. Divide so that it equals what you're trying to go to. So it's either going to be m equals I over a, or a equals I over m, or I equals m over a. One of these three, m, I, or a. But you can get to those by just doing basic algebra. The answer for this question is 1 micrometer. Alright, state the function of the structure labeled I. This is the structure labeled I right here. Now this is the cellular membrane. So what's the function of a cellular membrane? So a cellular membrane is used for the diffusion and separation of waste. Of waste and products that the cell needs to use. Now there's another definition. Just basically whatever a cell membrane uses. This is one of the examples. Deduce which type of cell is shown. Oh, so we just have to figure out what type of cell this is. I pretty much said it. It's a bacterial cell or a prokaryote. So we pretty much know that this is a prokaryote right off the bat. And how we know this? We need 4 reasons to how we know this. So reason number 1, the DNA right here, it's free. So if you see, this DNA is free. So free DNA. There also are no organelles. No organelles. There is no nucleus. And here, the DNA, if you look super closely, is circular. It's one big circle. So it starts here, it goes crazy, and then it ends off where it started. Just like a really big circle. So it's circular DNA. There you go. That's pretty much it. Starting now, I'm going to go through a lot of the answers, so basically everything that I just went over. And if you want to do that, a lot of people are going to skip to this point, so. Alright, question number 1. Outline the process of endocytosis, the process of moving large and a large amount of material into the cell. So, self-explanatory. 2. Explain the structure and properties of phospholipids, the fact that the hydrophobic and the hydrophilic properties, and they arrange themselves into a circle because of those properties. Number 3 is A, because it's exiting the cell. Annotate and label a diagram of the fluid mosaic. Now, I didn't really draw a detailed one here, but if I grab another piece of paper. This is super, I think is super really good that I diagrammed. So we have the phospholipid, these things. We have the peripheral protein here, integral proteins, carbohydrate, glycoprotein, cholesterol, and hydrophobic and philic tail. So you just basically draw one of these for 4. For 5, it's A, because water wants to be touching the polar substances, so these will have to be polar. 6, diffusion, going from high to low, no ATP, no energy, no protein. Facilitated diffusion, going from high to low, well, it needs the proteins, it does not need ATP. Active transport, going from low to high, needs proteins, needs ATP. Here, for 7, it's D, because it needs a concentration gradient in order to function. For 8, remember, cool raptors attack thick elephants regularly. Connect, recept, attack, attach, transport, enzymes, and recognition, connect two cells together, attach cells, transport materials, cells to be recognized. B, distinguish between active, passive diffusion, and facilitated diffusion. Looking at my notes here, this entire thing, want to take a picture, take it real quick. So, active transport uses ATP, goes from low to high, it needs a protein. Passive transport does not need ATP, needs proteins, it can stop. High to low. Diffusion does not need energy, no proteins, goes from high to low. Facilitated diffusion does not need energy, uses proteins, also goes from high to low. B. So 9 is B. The surface area to volume ratio, what limits? So we did this quite a bit. 10. There wasn't really a related diagram, you just look at this and then you do a diagram similar to how I had it here. Carbohydrates, integral protein, phospholipid. 11, state 1, 2, and 3. 1 is a cell membrane. 2 is a mesosome. This is to increase the surface area. I didn't really teach us that, but there you go. 3 is just a cell wall. B, calculate the actual length of the cell using the formula the actual over the A over IM. Getting that from the MIA. 1 micrometer. State the function of a cell, the structure labeled 1. So a cell membrane, what's the function of a cell membrane? Used to diffuse waste products out, products in, oxygen transport, all that good stuff. And then what type of cell is shown in here? It's a prokaryote. Reason is free DNA. There's no organelles, there's no nucleus, and the DNA is circular. There you go. That was basically a quick overview of this review. Now, if you have any questions, again, feel free to contact me. Just be like, yo, Rohit, what's up? Maybe during lunch. Yeah, alright. See you guys in the next video. Bye.